To understand modern agriculture, we must understand genetics. Scientists are developing new varieties of rice that will benefit small farmers in developing countries. Some, like golden rice, are created through genetic modification, where genes from other organisms other than rice were added to the rice DNA. Other times, scientists use information on rice DNA to decide which rice plants to breed. Understanding DNA is important to the beef industry as well. Ranchers breeding Red Angus bulls once had problems with birth defects in their calves, but scientists helped remedy this problem when they detected the gene causing this birth defect. Now, there's a genetic test available for ranchers to determine whether a cow or bull has this gene. The health of humans and their livestock are interdependent. Most all human epidemics, like smallpox, were caused by bacteria or viruses that originated in livestock and then evolved to infect humans. Even today, the same strains of bacteria can affect both humans and livestock, and some of them are becoming resistant to antibiotics. Scientists today study the DNA of bacteria in humans and livestock to see if the same bacteria can infect both. To understand much of modern agriculture, we must understand the structure of DNA and the biological process of reproduction. The twisting double helix structure of DNA is one of the most recognized images in the modern world, but not everyone understands exactly what the DNA components are. So first we will briefly discuss the structure of DNA. Then we will go to the laboratories at the Department of Biochemistry and Molecular Biology here at OSU to see how scientists determined that the same bacteria, S. aureus, were infecting both cattle and humans. Let's untwist our simple depiction of DNA so that it looks like a ladder and perform an autopsy of it. Before we identify its components, remember that all organisms are built according to the information contained in the DNA, and the structure of every organism's DNA is made from the same things. Just as a big or small ladder can be made from the same material, this model DNA could be the segment of a human, a worm, flower, or bacteria DNA. The sides of the DNA, usually called the backbone, are phosphate and deoxyribose, or sugar molecules. This backbone only provides structure to the DNA. It doesn't have any genetic information. The genetic information is contained in the steps of the ladder. You'll notice the steps are made of different colors, and there are only 4 colors: light blue, dark blue, pink, and red. These are our 4 nucleotides, and they go by the names of guanine, cytosine, adenine, and thymine. All organisms' DNA have only these same 4 nucleotides. What makes us different from the worm, plant, or bacteria is the length of our DNA and the sequence of the nucleotides. The length of our DNA could theoretically be the same as that of a plant, but so long as the sequence of the nucleotides is different, we will be built different. Rather than repeating the names of the nucleotides over and over, let's just identify them by their first letter: G, C, A, and T. Now I'm going to rotate the DNA horizontally and point out a pattern in the DNA. Notice that each step of the ladder contains 2 colors, which means each step is made up of 2 nucleotides that are bound together by hydrogen bonds. Notice the pattern though. Light blue is always connected with dark blue, meaning G is always matched with C. And pink is always matched with red, meaning A is always matched with T. What this means is that if we know one side of the DNA, we know the other side, allowing us to just concentrate on one side of the DNA. Finally, since we are concentrating on only one side of the DNA and all the information is contained in the sequence of the 4 nucleotides, we can represent DNA as a simple sequence of 4 letters: G, A, C, T, G, A, C, A, C, T. When discussing DNA, we can simply talk about it as a sequence of 4 letters, where the length of that sequence differs across organisms. That simple model of the DNA is misleading because it's so small compared to the enormity of an organism's actual DNA. So how big can an actual DNA be? Let's take the case of the simple bacteria S. aureus. S. aureus is an important bacteria because it can infect both animals and humans and is becoming resistant to antibiotics. I have the actual DNA of S. aureus here to show you. You can see this is a really big sheet of paper where all you'll see are different sequences of 4 letters, each 4 letter representing where each of the 4 nucleotides resides throughout the whole strand of the DNA. So this is the entire DNA of the S. aureus bacteria. And to print it off, I used 12-point font on paper 3.5 feet wide. But it contains 2.9 million letters. That means we need a lot of paper. It ended up going for 45 yards. Now, I would have liked to have been able to show you the human genome, but there's not enough room on this field. I don't have enough money to pay for the paper, because whereas this contains 2.9 million letters, the human genome contains 3 billion letters. That's over 1,000 times the size of this. So if I was to print out the whole genome, it wouldn't have taken 45 yards, it would have taken 45,000 yards. That's over 25 miles. And with genomes this large, whether it's the bacteria or the human, that's what gives rise to the diversity we see in life. That's what gives us the fungi and the fern, the mosquito and the monkey. This is why most all humans look different from one another, and all of us have different personalities. This is why my brother and I come from the same parents, yet look and think very different. You've been told throughout your whole life that you are special, and indeed you are, but you have the enormity of your DNA to thank for that. "45 yards, about the maximum distance from which a place kicker can score 3 points in football." But the human genome suffers no such limitations. And thus, the human proclivity for creativity, diversity, and genius seems about as infinite as the universe itself. In the next video, we're going to see how 2 biochemists at Oklahoma State University sought to determine whether the same S. aureus bacteria that were infecting people in hospitals could be found in the milk of dairy cattle. And to do this, they took samples of milk from dairy cattle, they grew the bacteria they found in it, and then observed its DNA to say, is that DNA the same DNA that we see in S. aureus bacteria? But they didn't have to check all 2.9 million letters. All they had to do was to see whether this 534 letter sequence was present in that DNA. If it was, then it was almost guaranteed it would be S. aureus.